We wanted to give you a brief idea of how we were seeing that webinar. It's flexible, so feel free to jump in anytime. We were thinking that the first half was roughly us presenting what we've done and what worked well, what worked not so well, and the lesson we learned at the end of the project. And in the second half, we'd love to talk to you about questions and discussion, or if there's specific points you wanted to explore further, we're available. So for the project, we've worked with use cases mostly. We found that that's what's working well for us. The use cases I was mostly focusing on myself is about biosamples and how can we validate data at scale. Biosamples is a samples metadata repository that is available here at EMBL-EBI in Kingston, UK. And what I mean by scale is at the moment we have, I think, just over 10 million data points. So 10 million samples in our database, which are shared with our partner, as you can see at the bottom of the screen, through INSDC. We're exchanging our sample information with DPG, which is a database in Japan, and NCBI in the US. So it's very much of a worldwide coordination for sample metadata. You can see that locally at EBI, so on the right-hand side, biosamples link to several of the EMBL-EBI archives, so Magnify if you're interested in metagenomics, EuroPMC for literature information, ENA for sequencing, EGA if you're doing human controlled access, and so on, RExpress, EVA, and so on. And the data in biosamples on the left-hand side comes from a variety of projects. There's the research community as a whole, but there's also specific partners such as the EBISC project, HIPSCI for stem cell, FANG for farm animal genomics, PDX if you're looking at patient-derived xenografts in mouse. So all those projects have obviously a variety of data that is coming in through our data submission portal, and we have high interest in validating that data as it comes in. And on the next slide, why do we want to validate that data? Because the goal at the end is to be able to link to all those archives. So if you're a researcher and you're doing some sort of assay experiment on a set of samples, we'd like you to be able to come to Biosample and say, okay, there's some data in ENA, there's some data in RExpress, and by entering via the Biosamples database, I can collect all the information at EMBL-EBI on the samples I'm interested in. And to do that successfully, we require a good infrastructure. I'm not going today to tell you about the backend on the Biosamples database, but it's great. And it also requires good metadata, and I think that's very much what we are concerned with today. And good metadata is about the ability to find the data, to interoperable data, and be able to reuse the data once we actually acquired it. And the problem we have today, specifically here is the example of Biosample, is data is very diverse. I told you it's coming from a variety of projects, and you can see here we have just on the right-hand side a word cloud of the attributes in Biosamples. Unsurprisingly, organism, very prominent, very common. We found that pretty much all our submitters could agree on what an organism is and represent it consistently. But then you can look at specific example where you have sample type, sample title with an underscore, collection date with a gigantic typo in there, which is 900,000 of a sample. This is not great. And if we could improve the quality of the data as it comes in by validating the data as it comes in, then we think we can lower the cost of data curation on our side by having the data harmonized at submission time. The graph at the bottom right shows you the frequency of attributes in Biosample. So the big peak on the left That's organism, present pretty much everywhere. And then you have that very long tail of attributes that are used a couple times in the whole database. Some of them are in between, gender, female, things like that. And again, we're trying to find the right data. So this is an example of use cases from the Global Alliance for Genomics and Health. We really want to be able to query biosample to say, hey, can you give me all the sample of people that are male, over 70 years old, and have lung cancer? Can you find all individuals that have an AML diagnosis and also Down syndrome, and so on and so on? Find me the data that has been consented for specific research purposes. So we're really trying to do that, and doing that today on biosamples is very hard because we don't have harmonization and standardization, validation on what a diagnosis field is, on what the gender field is, on how we represent geographical location or consent codes for data use. So on the next slide, I wanted to tell you about the way we've thought about doing that validation. And when the validation study group came together, we looked at several options, and we decided that JSON schema was really the way we wanted to go for us. The way JSON schema works, on the left-hand side, you have biological schemas, so you can define, this is how a sample is represented for me. And this is going to say, sample has a name, there's a taxon, there's the age of your donor, there's some sort of unit for the age, and so on and so on. And then when the instance data on the right-hand side comes in, you can validate automatically that data against the schema you declared. So here we are saying the tissue is liver on the right, and you can see on the left, tissue needs to be some sort of string for that value. And that can be automatically validated as it comes in. Why JSON schema? On the next slide, we didn't choose JSON schema totally randomly. We looked at what other people were doing and what we could reuse, and people specifically we work with, and what we could leverage and have something that would be interoperable as well. We work closely with the Human Cell Atlas, HCA. I mentioned the FANG animal genomic project earlier on. ENCODE and other archives at EBI, such as the European Nucleotide Archive, BioStudies, and of course Biosamples, are also using JSON schema. So that was really a clear choice for us. Taking together the project that we're using it and the tooling support for JSON schema was really a critical factor in our case. We moved forward with the decision, and I'm very happy to mention that at the end of the project, we've managed to create a validator that is JSON schema based. It's currently used in production by the Human Cell Atlas, Biosamples, and the submission portal at EBI for general data submission. And this one lives under the ELIXIR organization GitHub repository. So it's all open, free to reuse. You can fork the repository, you can add your own local extension. Feel free to take it for a spin. Any problem, let us know. It's been working well for us. So we have metadata schema that are defined using JSON schema, and then we're able to, as I showed earlier, automatically validate the instance data against those schemas. Interestingly, on the next slide, we found out some features that were missing from JSON schema. Many of you on the call will know that sometimes we need to be able to validate things against an ontology, for example. So if I want to say this value of disease, in this example, type 2 diabetes mellitus, should be a term coming from the disease ontology, we can do that. No? So we can specify an ontology ID, and we have a little piece of software which is part of that validator, which is going to make sure that the disease is part of the disease ontology tree, and that term exists. And this is something that has been extremely useful, specifically for the human cell atlas, and it's proven so useful that the JSON schema developer are actually adding that type of validation in the new version of JSON schema, the draft 8, that's coming out sometime. So this has been really working very well, and we're very happy with that specific outcome. On the next slide, we were then looking at data exchange. So I mentioned we're exchanging data with, for example, our Japanese and US colleagues. Another aspect we were really interested in is enriching our existing metadata. On the left, we have a screenshot from the biosamples website. So this is a sample that we have actually coming in from NCBI with a bunch of key value pairs for attributes. On the right-hand side, a colleague from the Marine Metagenomics Reference Database, MARREF, and that's the URL to access that record. They are very interested in marine metagenomics, and they have recreated a bunch of samples using publication and additional knowledge. And you can see the difference between the two records really striking. We have something in biosamples, which is something minimal, but on the MARREF side, there's really way more metadata, and we'd really want to be able to get that automatically from them and enrich our metadata locally. And we're looking at building that on the next slide, building that system for metadata exchange between the curator and the existing archive with support from the ELIXIR Clearinghouse project and relying on another ELIXIR project BioSchema. And at each step, we would use the validation that we've developed. So the JSON schema-based validation in the case of biosamples and the clearinghouse, and also the bioschema-based validation that our colleague Alasdair has developed as part of this project. So that leads nicely into the plant use case that Frederic will describe. So I just told you about metagenomics. We were also working with the plant community, and interestingly for plants, there's currently no public repository for plant phenotyping data, and Frederic and his colleagues were very interested in the option to use biosamples to host and validate that data. Passing over to Frederic now.
Thank you, Melanie. As Melanie mentioned, we have been really focusing on use cases to make these validation in real life and not some abstract discussion. And we have teamed up with the plant science community within ELIXIR, which has as a goal to define and implement standards for representing phenotypic data and its metadata across the plant science domain, and develop a federated database and the accompanying web portals to discover this phenotypic but also genotypic and other related data. To actually make sure that we can demonstrate this, we have also put some effort in annotating and submitting exemplar data sets to both resources at EBI as to the infrastructure that we have been developing. And obviously we want to spread this way of working, the best practices and the tools within the plant community. To achieve all this, we have worked with a number of technologies, which I will briefly introduce. First of all, MyAPI is the minimum information standard about the plant phenotyping experiment. This is a community-driven project which was started in a FP7 project transplant, and where ELIXIR has continued developing this standard, aiming to harmonize data from plant phenotyping experiments across the globe. And then conceptually, this is a checklist of metadata that is required to describe a plant phenotyping experiment with the software tools to validate, store, disseminate this data. On the links on the slide, you can find all information about this international consortium. So this is where we develop the metadata standards that we need. We also need a way of sharing data because, as Melanie said, there is no centralized repository for plant phenotyping data, but there are a lot of databases in institutes that contain this data. So when looking for a way of sharing such phenotyping data, we came across the Breeding API project, which tries to develop a RESTful specification to enable interoperability among plant breeding databases. This turned out that also for academic institutes doing plant work, being it plant breeding or beyond, this is a very interesting approach and a very interesting project. So we teamed up with the BRAPI project to develop this standard interface to access plant phenotyping and genotyping data. And again, we did this in a shared, open, community-driven way to develop this standard. In the plant community, ISA tools are often used to share data, and we wanted to take advantage of this existing community. ISA is an open source framework and an accompanying suite of tools to manage life science, environmental, biomedical experiments, and is really focused on the description of metadata of experiments to make the data, the discoveries, reproducible and reusable across domains. ISA stands for Investigation, Study, Assay, and this is a logical separation of the different metadata levels within an experiment. So you have the project level, there's a study or kind of research line that is a part of that, and then in each study you can have several assays, which are the actual measurements that you do. ISA has a whole ecosystem, and you can find more information on the ISA tools or ISA Commons websites. Now I will briefly go how we brought this all together in this data validation study. So on the left hand, we have breeding API endpoints. This is a result of the Accelerate project, where across Europe we have implemented the breeding API on top of existing databases containing plant phenotyping. We have done this in our institute in Belgium in the PIPA database, but colleagues in France, the Netherlands, Portugal, Slovenia, UK have been implementing this, and we are even broadening this beyond towards, for example, Germany now. On the right-hand side, we have MyAPI, where we have this checklist of metadata, and within the Accelerate project, we have fixed version 1.1 of this checklist, and this has been formalized in the plant phenotyping experiment ontology, which is, of course, very tightly linked. And we are also working on a JSON schema and JSON LD schema representation of this metadata standard. To link this together, we want to use ISA tools. So on the first side, to get access to the data, we have developed a script that takes breeding API endpoints and provides this data and metadata in an ISA object way. On the metadata side, we have integrated the MyAPI standard using the ISA creator configurator to a configuration file that is appropriate for ISA. If we now take these two things together, we can take the data from the breeding API endpoint, take the configuration, and using the ISA creator functionality, create the ISA tab files, which are the files that contain the complete description in an ISA compliant way of the experiments. We did this in an automated way using the ISA API, and this ISA API also allows us to validate these ISA tab files. Biosamples is compatible with ISA and is able to accept submissions through this. So this is a very nice link to submission at EBI, and will allow us to link up the phenotyping data with transcriptomics data or genotypic data that is available at EBI services. This is one of the reasons why we chose to go with ISA. There's a number of other things that are related. So we are now also starting work with bioschemas to represent MyAPI in there. And as Melanie already alluded upon, we are collaborating with Alice there and valid data to use the RDF way of working with shape expressions to also be able to validate this way of sharing data. On the BRAPI endpoint side, there's also an aspect of validation, which I won't go into detail, but we need to be able to validate that the endpoints are actually conforming to the BRAPI standard. And for that, we have, again, a BRAPI JSON schema and a BRAPI validator called Brava, developed by IPK, which allows us to validate the conformance of the BRAPI endpoints. An additional point I want to make around the ISA tab and why we chose this is that this is also a format that researchers that do not have access to a BRAPI endpoint or have the resources to set this up, they can make this themselves using spreadsheet softwares. And this way we can be inclusive and we don't rely on high-tech solutions to get researchers in. The next steps in this project are to finalize and fine-tune the implementation. We are almost there. We are making the final adjustments to make sure that the BRAPI to ISA is MyAPI compliant. And we are now also looking into getting the ISA JSON support so we can tap into all the work done in, for example, human cell line class around this JSON validator. Next steps, as mentioned, are JSON LD, where we want to link with bioschemas. And on the somewhat longer term, we are developing the Breeding API 2.0 version, where we will improve MyAPI compliance even further. To link this all to what projects we are doing in the future, there is a plant community-led implementation study that will start 1st of June, which aims to provide a toolbox to submit data to the EVA, the European Variant Archive, so the genotyping data, and enrich this with the necessary metadata such as the multi-crop passport description and the MyAPI plant phenotyping description. Again, this implies submission of data, so we need validation, and we will be relying on the software and the infrastructure we have been developing in this project. The EOSC-Life project, which is the biomedical cluster project for the European Open Science Cloud, there is a demonstrator on plants where we want to integrate genomics, proteomics, metabolomics, phenotyping data that is spread across databases both in Europe as local. And so we have teamed up with Fairdom to solve the what we call first mile problem. A lot of researchers have data in-house and they need to manage this also, but often they do not comply to any metadata standards because they don't have the infrastructure to enable this. With Fairdom, we want to enable this local data management according to the metadata specifications of the deposition databases, core data resources, and the plant phenotyping infrastructure that we've built to get all the metadata from the moment the data is generated rather than as an afterthought. Again, validation will be important to ensure that the submission is done in an appropriate way. I also want to touch upon another implementation study which we've added as a use case, which is on the verification of genomic data tracks. Our colleagues in Spain and Norway have developed a comprehensive fair genomic data tracks metadata model, and they have been using JSON schema to validate this, and in collaboration with people at EBI, they have adopted the word there and extended it towards validation of ontology terms, validation of the consistency between free text and ontologies, and also validation of the complex URLs in identifiers.org. So this aligns very well with what we have already been discussing. They have extended this work done in human cell atlas and the data validation implementation. And on the Spanish side, this really links into...
Sorry, Frederick, about this. We'll quickly tell David to mute.
Yeah.
David, actually, we can mute him. All right, go ahead.
Okay, so from our Spanish colleagues, this really ties into the Open eBench that they are developing. This is a platform for benchmarking of workflows, and there's a lot of validation aspects in this. First of all, there's a benchmarking metadata model that needs to be combined, but also you need input data for the workflow, which needs to be validated both on format and identifiers, and also around this identifiers, as already said, these queries needs to be checked for compliance. With that, I want to wrap up with the lessons learned. Once you start looking, validation seems to be everywhere, and more often than not, this is a very neglected aspect where people assume that data going in or metadata provided is compliant to a specific standard, which is unfortunately often not the case. So if you start really looking into this and implementing this in the nitty-gritty details, this starts to become complex, so it's important to carefully determine and safeguard your scope, what you want to do. And we've experienced that the use case driven approach is really key to this, because there's a lot of details that are specific for domains that you can only discover using these exemplars as a driver, and this will really allow you to make appropriate software and developments to actually solve the problem. We have also been thinking about sustainability of this data validation beyond the implementation study. Everything we have been doing was open development in GitHub, both for the validation tools as for the standards that we are developing. This is a hallmark of ELIXIR, that we try to do everything in open development. And I think we have succeeded in getting adoption and alignment and improvement of the tools across different domains, as demonstrated by the different use cases that we've presented today. And finally, this data validation implementation study is really turned out to be a driver to bring the community together. We have been implementing standards as a collaborative effort. We met several times face-to-face, and think we've done a lot of the work in the ELIXIR Biohackathon in November 2018 in Paris, where we really got the right people together and started the actual development in all its nitty-gritty details for real. And this multidisciplinary team that we have is really key to the success of this data validation implementation study. We need experts in all aspects, from how you represent things in ISA, on ontologies, plant phenotyping data, the genomic tracks, the human cell atlas. In all these things, you need experts that are in the field, that know all the details, to really make this work out and then be a success. So with that, I want to thank you. You can contact us on this mailing list that we have for the validation study, and I really want to thank all the people involved. There's a lot of people that have contributed to this, and there's a number of projects and consortia that have been contributing to validation study